In chapter 3, the focus is on cell structures, and the question is also asked, how is the structure of a cell related to its function? So of course, if you look at the individual parts in the cell, they're each going to have their own function. Also, we can look at how do cell malfunctions result in pathologies. So if something goes wrong with that functioning of the cell, what will be the result? Now, when we study cells, of course, we need a microscope. The study of cells is referred to as cytology. Now, I made a little point there at the first of this slide. You'll see I said to review the different types of microscopes and how they are used to view cells. That is not really required for a survey of biology course, but just know that there are different kinds of microscopes that are used depending on how much detail you need to be able to identify on a cell or inside of a cell even. Now, a person who studies cells to determine if they are abnormal would be called a cytotechnologist, and such a person would be working in the lab with microscopes and looking at these cells, comparing normal with abnormal. For example, at the top here you'll see a normal pap smear. This is a sample of cells from the cervix of a lady, and you can see this is what those types of epithelial cells typically look like. But if they're abnormal, you can see that they sort of clump together and they have a change in their shape and also size. Now, cell theory is a very strong theory. In fact, based upon thousands of observations over hundreds of years. And we credit Robert Hooke for being the first person to look through a microscope and record cells. In fact, he was looking at a piece of cork and he noticed that there was a specific arrangement to these cells under the microscope. Now, we also credit Antony van Leeuwenhoek because he was the first scientist who actually saw little, what he called animalcules swimming around in a sample of pond water. He was curious to know whether there would be living things inside of water, so he basically just took some water samples from after a rainstorm and he put it under his microscope and he saw these little things swimming around. Now, two other scientists also deserve credit here for their role in developing the cell theory, and they are Schleiden and Schwann, and both of those had done many observations, and they looked also at the data and observations from other scientists and put together what we today refer as the cell theory. Now, the first point in the cell theory is that all living things are composed of cells, because we know from looking previously at the characteristics of life that a cell is the smallest living unit. So if something is alive, it has to be consisting of cells. Then cells always come from other cells. You do not have cells just spontaneously arrive or arise out of nothing. They have to come from previous cells. And thirdly, like we said, the cell is considered the basic unit of life. Now, if we compare cell sizes with one another, look at this interesting chart. Most of you have seen a chicken egg. Maybe you've had that for breakfast. You can, of course, see that with the naked eye. And an ostrich egg is even bigger. You may have seen frog eggs maybe in a pond. You know, they are observable with the eye. But as soon as you get down to this scale, the size of a human egg, this is small. This is about the size of the top of a pin, you know, the human egg. And then for anything smaller than that, you typically need a microscope to see the structure and the detail of an animal and a plant cell, and of course bacteria, which are even smaller. And then all of these structures inside of the cell obviously are going to be smaller than the cell. So relatively, you'll see that there are some here that you can see with the naked eye. Some you would need a simple light microscope like what we have in the lab here. And then, of course, if you want to see these smaller parts, you would need a strong, high-powered electron microscope. Even to see some of the viruses that we have described, you would need that type of electron microscope. Now, Now, why is it important to know about cell size? Well, without getting into too much of the math, scientists know that surface area and volume are limiting factors for cell size. So what does that mean? Well, think about it this way. If the cell grows too large, the membrane, that covering around it, simply can't transport enough materials to the innermost portion of the cell. Think about this, how long will it take for things to reach the center part of the cell? If the cell gets too big, it's just going to take a very long time to get to the center of that cell. So it makes sense if you have a smaller cell that, for example, if oxygen needs to get into that cell, that it would reach the inner part of the cell much faster if the cell is smaller. So really here, what we look at is the surface area to volume ratio. And here you see it's 1 to 5 to 1, 1.5 to 1 rather, 3 to 1 and 6 to 1. And if you have a higher ratio that you see here in the smaller cell, this is what you strive for. The higher that surface area to volume ratio, the better that cell would be able to deliver things to the middle of the cell. Now, prokaryotes have a really good ratio. They of course are bacteria, for example, and they have some unique structures that allow them to survive in their habitat. I want you to notice the first striking thing. Inside of such a bacterial cell, you have a region called a nucleoid. Now, this is not the same as a nucleus. Maybe you should underline that just to make that stand out. A nucleoid is not the same as a nucleus, but it is considered an area in which you find the DNA. So somewhere in there you're going to find in that nucleoid region the DNA. And then of course around it you see labeled here there is some ribosomes. In the cytosol, this light blue area here is the cytoplasm, or what we call the cytosol. And then of course you're going to have a plasma membrane, a cell membrane, that first membrane there around the cytosol, and on the outside of that a cell wall. And what makes bacteria unique is that they can even have a capsule on their outer edge, and that's what makes it so hard to kill bacteria, because they have this reinforced capsule and cell wall. Also, other interesting things, you note that they have pili and they have flagella. They may have more. In this picture there's just one flagellum. Pili help them stick to surfaces and flagellum help them move. So if we compare a prokaryote to a eukaryote, we just said that a prokaryote has a membrane. It has DNA but not in a nucleus. It has ribosomes. But note here it has no membrane-bound organelles, whereas eukaryotes do. And that is the difference really between a pro and a eukaryote. Pro means before nucleus. Eu means true nucleus. But the other difference is here, very important. Eukaryotes have membrane-bound organelles. So they are a little bit more advanced than prokaryotes. So let's take a look at eukaryotic cells. Their main features are that they have a nucleus. Remember, we said that that's what sets them apart from a prokaryote. They have this nucleus, usually in the center, somewhere in the cell, and inside there you're going to find some chromosomes, labeled here chromatin, and that's where the DNA is found. You're going to have various other organelles. Look, we did not see all of these other organelles in a prokaryote, right? We said that that's a function, rather a characteristic that a eukaryotic cell has. It has these membrane-bound organelles. And of course there is also some cytosol, because that is the matrix, the liquid interior in which all of these little organelles are contained in. Okay, so you need to know the basic structure of a eukaryotic cell. You see things labeled here, and I think we're going to go through them one by one, make that stand out a little bit more to you as we go through these slides. I first just want to say something about the endosymbiosis theory. There is a theory that provides a possible, take note of that, a possible explanation for the origin of eukaryotic cells. Particularly how maybe these mitochondria and chloroplasts came to be inside of a cell. So most of these organelles you have in the cell probably, according to this theory, originated from the folding of the plasma membrane. For example, here you can see if the plasma membrane starts to infold, you could have those migrate and form around the nucleus and nuclear membrane system. So they also speculate that maybe these mitochondria and chloroplasts were taken up by these cells, that they did not have them originally, but in some way they took them up and they survived inside of the cell. They were not killed or digested, and now they have a function inside of that cell. Okay, if we look at the outside of the cell, there is a very special layer called the plasma membrane. It is actually consisting of phospholipids, these little molecules you learned about in the previous chapter. Phospholipids can also have some proteins embedded in them. So that means they are not going to be on their own. You're going to have the phospholipid bilayer. So there you see one phospholipid, and there you see some more, one pointing in the opposite direction. But look here, you can see that there may be all sorts of proteins. These blue things you see embedded here are proteins inside of this phospholipid bilayer. Now those proteins are going to make it possible for the cell to perform many different functions. You may even have some other structures associated with a phospholipid bilayer, maybe cholesterol molecules, and maybe some filaments of the cytoskeleton to make the membrane stronger. Now this plasma membrane is a barrier. It is going to protect the cell because you don't want all sorts of things just to enter the cell. You want to protect the inner working of the cell. So it will allow some things to go through. You see there's some channels, but it's going to control how those things move through. The nucleus, let's focus there for a moment. We said is the central part of the cell. It actually contains the DNA, which is in the chromatin, which is in the chromosomes. And the function of the DNA, of course, is to store information. Now, often I see also the function of the nucleus described as a control function, and that is true. Of course, the information is going to be used to control all the inner workings of the cell. So around the nucleus, you're going to have an envelope to protect it, and inside there's going to be some nucleoplasm. It's consisting of the same sort of liquid matrix that the cytosol or cytoplasm is made up of. So if you had to label a nucleus, inside there's actually a core called the nucleolus, and then you have the nuclear envelope number 2 here, and then around that you have the system called the endoplasmic reticulum. We'll speak more about that in a moment. A on this diagram is labeled DNA or chromatin. Now ribosomes are also found in the cell, and ribosomes are the site for protein synthesis. These ribosomes are all throughout the cytoplasm. They may be free in the cytoplasm, and then we refer to them as free ribosomes, or they may be attached to the endoplasmic reticulum. Jumping back here, you'll see that is the membrane system. So look again here, you'll see all these little yellow dots here. These are ribosomes attached to the endomembrane system. Now, ribosomes, we said, are sort of these little factories that make proteins. And we're going to learn more about how they make the proteins later in this class. But for now, it's just sufficient to know that a molecule called messenger or mRNA is going to bring in a code. And then this ribosome will read the code. And based upon this code, it's going to produce a specific peptide, which is another term we use for protein. So this is the growing protein. Here you see it consists of little molecules of amino acids, which you learned about last week. Those are the building blocks for proteins. Now another molecule is involved here called tRNA, transfer RNA. It actually helps to read the code and to bring in the correct amino acids to build that protein. Mitochondria, you learned probably when you were in high school, middle school, that this is the powerhouse of the cell. So it is going to provide energy, and that energy is in the form of ATP. Now, specifically, they are responsible for the process called cellular respiration. We will not study that process in a whole lot of detail. We'll go through the basics, but in a nutshell, that is the process during which ATP energy is produced. So you'll see a mitochondrion has this outer membrane, and then it has this inner membrane that folds in on itself. Those folds are called cristae. And to the inside of those folds, the area is called the matrix. And in the matrix, you see also some strands of DNA. In fact, mitochondria has their own DNA that is separate from the cell's DNA. And often this DNA is used in crime scene analysis because this actually lasts longer than regular DNA in case of, decay occurring. So what happens if your mitochondria does not work? Well, you can develop a mitochondrial disease. I posted here for you an interesting video to go look at, a mitochondrial disease, and it really is a devastating type of disease. There's many kinds, and for many of them we do not have treatment or cures. So it's very important that scientists keep studying mitochondrial DNA and understand how mitochondria function in order to help these individuals maybe, one day cure the disease. But I have a question here for you. Consider that mitochondria have their own DNA separate from the parental DNA. How could you cure this disease? That's very interesting, Think about that as you watch this video. And we can certainly talk more about this when we cover DNA in class later this semester. So, be able to label a mitochondria. These are the parts. I gave you a practice diagram here again. Can you label the inner and outer membrane, the cristae, and the matrix inside of the cristae? That would be important to be able to identify. All right. So I hope you looked at that little video of the mitochondrial disorders. If you're interested in more, I'll put some more information here. In fact, over the years, I've had several students in my classes tell me that they have family members with mitochondrial disease. So I know this is something that may be near and dear to some of you. Maybe you have family members that suffer from some of these disorders. So these mutations, either in the mitochondria's DNA or in the nucleus's DNA, is what leads to these types of disorders. I want to just mention something here quickly. It is, like I said, an advanced disorder. Scientists try their best to understand the working of the molecules, and in this case, during cellular respiration, there are many enzymes involved. At any point, something can go wrong with these enzymes. So, that's going to influence how many ATP molecules that person can make. And that's why not any two people with a mitochondrial disease have the same exact symptoms, because it may be for one person that this first enzyme is affected, for another one it may be the second to last enzyme. So it is actually quite a complex disorder that we're referring to here. Okay, I want to move on to peroxisomes and lysosomes. These are organelles in the cell that contains enzymes that help to keep the cell functioning properly. Peroxisomes specifically has an enzyme called catalase that breaks down hydrogen peroxide. You don't want to have too much hydrogen peroxide in your cell. That is a free, radical. It's going to be an oxidative process that occurs breaking down stuff that doesn't or shouldn't be broken down. So you want to get rid of as much hydrogen peroxide as you can. And the lysosomes, they are considered our garbage disposal organelles because they help to break down substances and they also help with digesting food. All right. Now, lysosomes contain an enzyme called hex-A that helps to digest GM2, for example. So say something goes wrong in a human and there is a mutation that causes hex-A not to be produced. well, we know that this is actually called Tay-Sachs syndrome. It's a genetic disorder. So if that child does not have any hex-A, GM2 will build up in the cell and eventually kill it. We do not have a cure for Tay-Sachs disease, and there's a lot of research going into this field as well. So this is just a sort of application to help you understand why should we care about these little organelles. Why should we learn about their function? It really does help scientists in the long run understand what goes wrong in case of a function that is negatively impacted. Now, vacuoles are used for storage and transport. So they're kind of like the storage containers of the cell. In plant cells specifically, they store water inside of a large vacuole called a central vacuole. So you usually see it depicted on a plant cell diagram sort of towards the middle of the cell, a huge central vacuole. Animals and protists have vacuoles that can store nutrients. So those would be called food vacuoles, and you can see those here in green on this diagram. And of course, here, this contractile vacuole looks a little bit different. This can store water, and it can also help to get rid of extra water. So it will actually squeeze out the water out of this little star-shaped organelle. Right, centrioles and centrosomes are also important. These are what help to move chromosomes around in the cell during cell division. So here we have those centrioles arranged in a centrosome configuration. So if you look at each of them individually, they are consisting of centrioles, but the whole thing in total, so this would be one centrosome pair. That's what they're known as. They are only found in animal cells. Scientists have not identified or seen them in plant cells. So you'll see here in this picture, it is depicting a stage of cell division, which we'll learn more about later. These little chromosomes attach to the spindles here, and then the centrosomes move them along these spindles to the opposite ends during that cell division process. Right, moving on to chloroplasts. These we only find in plant cells. Of course, they are responsible for the reactions of photosynthesis. You may have heard of this process. Typically, this is a process that you learn about also in high school, middle school, and you may also know that they have their own DNA. They are often studied in a lot of detail, and there's a lot we know about the chemical reactions. So we're going to briefly go over those reactions when we cover the photosynthesis chapter. You'll see that there are little stacks inside of a chloroplast. Kind of looks like stacks of pennies on top of each other. Those are called granums or graina. And each individual one is a thylakoid, consisting of a thylakoid membrane and a lumen. The yellow, space inside is a lumen. And then note that the graina are connected to each other with the flat green areas here called lamella. Also, you'll see it is surrounded by stroma. Stroma has the same consistency as cytosol or cytoplasm. And there may be some ribosomes floating in here as well, because there are some proteins that need to be manufactured for the process of photosynthesis to take place. Around the cell wall, you find around the plant cell, you find a cell wall, and also around the bacterial cell. Remember, we mentioned that when we started our discussion off with prokaryotes. So it is a very rigid structure. It's made of cellulose, a strong type of carbohydrate. So that makes it really difficult for it to be digested. Plants with cellulose are considered high in fiber. Maybe you've heard that terminology used. And these types of animals that eat plants usually have extra help in the form of little bacteria that live in their gut to help them digest the cellulose. Okay, so at this point, I think what would be helpful for you to do is to go and review what we've discussed so far. Maybe pause the video here and use this table. Check off whether bacteria have these structures, animal and plant cells have them. I think this is a good way to compare and to remember what does bacteria, animal, plant cells have in common and what do we see their differences are. And when you're done with that, you can watch the rest of the video. Right, let's get back to the endomembrane system. We briefly mentioned that when we spoke about the nucleus. This membrane system around the nucleus is called the endomembrane system, and it takes part in processing, packaging, and secreting process. So you can actually think of this as sort of as a processing plant where things are being made. They have to be transported somewhere, so they have to be packaged, almost like you would do at a post office, and then exported. So we said previously the ribosomes make proteins. So there you see rough rough endoplasmic reticulum. That means there's lots of ribosomes on there, and they are of course busy making proteins. So zoom in here, you'll see there are some proteins leaving the endomembrane system, and next they will go to an organelle called the Golgi apparatus, sometimes referred to as the Golgi complex. And now those proteins are packaged and they go through this Golgi apparatus that kind of looks like a stack of pancakes, and then they are processed and then they are shipped out of the cell. Note that they go into these little vesicles before they go into the Golgi, and then they're packaged again in a little vesicle before they go out. The part of the Golgi where they enter is called the cis phase, and the part where they exit is called the trans phase. That's just a way to distinguish between where they enter and where they exit. All right. Now just to tie those two functions together, here on the left, remember we said the ribosomes are like the little protein factories or synthesizers of the cell. They make the proteins, and then the Golgi have to take these proteins somewhere where they are going to be used. So it is a good analogy to use here if you say the Golgi apparatus is like the post office of the cell to go and deliver those packaged proteins to the specific places they need to go perform their function. Right, these little vesicles that we mentioned, they can fuse with the cell membrane and they can transport materials out of the cell. So they're going to move from the trans phase towards the cell membrane and out of the cell. And of course, inside of them, they have those little proteins that were manufactured. Right, so this is also helpful, I think, in preparing for your exam, where you probably get some diagrams to label. See if you can label the parts, starting here, with the nucleus in the cell. See if you can figure out what is happening here. This illustrates the movement of these little vesicles throughout the endomembrane system after they've also been manufactured on the ribosomes. Number 1 would be the nucleus. 2 would be a pore in the nuclear envelope. 3 would be the rough endoplasmic reticulum. Number 4 would be some smooth endoplasmic reticulum. Maybe you don't see the difference right away, but look here on this part, there are not any purple ribosomes on top of the endomembrane system. Here you have these little purple ribosomes, right? Number 5 is the ribosomes. So if they don't have any, we call that smooth ER. All right, number 6 would be little proteins being made. Those little green dots, they next are packaged into a little vesicle and they go to the Golgi apparatus, the cis phase. They move through the Golgi. These little membranes in the Golgi are labeled cisternae, and then they are packaged in vesicles again on the Golgi's transphase. All right, so you can review by covering the names here on the side and see if you can label those parts. Same here. Use this diagram, see if you can review, because the diagram that I'm going to give you in your exam is probably not exactly the same as the one here in the PowerPoint. All right. There's also some videos available on YouTube. If you just Google the endomembrane system, for example, there's some pretty cool animations to show you how these little vesicles move through the Golgi after they are delivered by the endomembrane system. Okay, I want to next talk about the cytoskeleton. This is a network of fibers that give support and structure to the cell. There's many kinds of cytoskeletal elements. We have microfilaments, microtubules, intermediate filaments, and they help with moving and maintaining shape. In specifically these microtubules, we see flagella and cilia are composed mainly of microtubules. So if you go and you zoom into the microscopic structure of a cilium or a flagellum, like in, for example, this is a sperm down here. If you go look inside of this flagellum tail area, you'll see these little microtubules inside, and that's what helps it to move. These are actually little proteins in there that help the flagellum to move. Now, you may think, well, why should I know about cilia? Why should I know about flagella? Well, let me give you an example of why it's important to know about cilia. If cilia are not created normally, you can end up again with a disorder. For example, primary ciliary dyskinesia. So if it is mutated, and there can be different mutations, of course, it's not going to have the proteins arranged in a normal fashion. It's going to be abnormal, and then these cilia cannot function properly. They're not moving as they're supposed to be moving. So these types of individuals that have PCD, they are more prone to sinus infections, pneumonia, and runny nose. And of course, in the reproductive system, you also have some cilia. So probably those females will have some infertility issues because their eggs cannot move along in the fallopian tubes as they are supposed to. So there's some other individuals that have maybe situs inversus, where it's an even more problematic scenario where the cilia is not functioning properly. Okay, let's look next at how cells are connected to each other. We have things called gap junctions. Those are little gated channels, just like the word says gap. So it's a little channel or a gate between two cells that allows things to move in and out of a cell. You also have between plant cells things called plasmodesmata that help plant cells to exchange things with each other. You also have desmosomes, which are little adhesion proteins that help to join cells together, and tight junctions. Now, tight junctions are what the name says. They're tight. They tightly connect cells together, whereas a desmosome, you can see there's more space in between the two cells. So I would say this is an easy way to distinguish them. If you see it sort of look like it's stitched together with a pattern here, it would be a tight junction, whereas if it looks more stringy, thread-like, it's probably an adhesion junction or a desmosome. All right, so I've come to the end of my discussion about all the organelles. I think labeling, practicing, identifying the parts of the cell is really what you should be focusing on. And can you then, after you have labeled it, give the function of that structure? So use this slide, this diagram, as in practice. There's another one with a little bit more to label and identify. See if you can identify those parts. And then I also put another one here for you